Bismillahirrahmanirrahim. My name is Dr. Sagheer Ahmad, and today we are going to discuss the first step of oxidative phosphorylation, our first step of respiratory process. What is the name of that process? That is glycolysis. The breakdown of glucose, that is what the first part of a respiratory process in case of oxidative respiration in the presence of oxygen. Okay, now let's start. Glycolysis, the breakdown of glucose in the aerobic respiration. It is the first step. Okay, suppose cell has taken up the glucose molecule. Okay, here is what the glucose molecule 1 2 3 4 5 6 6. It is 6 carbon compound, and it has reached into the cell. Okay, this is that glucose molecule. Okay, now let's start. All these reactions do occur here in cytosol. You can see that the site of glycolysis is cytosol. Okay, how do these reactions occur? Let us start. Suppose we have a glucose molecule. This glucose molecule would undergo a biochemical reaction like this. This is your alpha D glucose. Okay, it will undergo a biochemical reaction like this. One ATP is used in this reaction, and it gives one phosphate, and it becomes ADP. Okay, well, one magnesium ion is used here. Okay, the enzyme used in this reaction is called what? Hexokinase. The enzyme is known as what? Hexokinase. Okay. What is formed at the end of reaction? What is the product? You see, this is glucose 6-phosphate. Why do we call it glucose 6-phosphate? Because this is carbon number 1, this is 2, 3, 4, 5, and 6. And the phosphate has attached itself with carbon number 6. So we call it what? Glucose 6-phosphate. This is the first reaction in glycolysis, right? Now, what next? Glucose 6-phosphate undergoes the next reaction and forms what? Fructose 6-phosphate. Fructose 6-phosphate. This fructose 6-phosphate would undergo the next reaction and it will form what? This is what? Fructose 6-phosphate. Okay. The enzyme used here is phosphohexoisomerase, because fructose is the isomer of glucose. That's why phosphohexoisomerase. Okay. In the next reaction, what happens? Another ATP is used. Another ATP is used. This is, say, ATP. ADP would be received from this ATP. Again, magnesium ion is used here, okay, as activator, and the molecule which we will have, that is what? Fructose 1,6 fructose 1,6 biphosphate. So that ATP which we have received from here, that has been attached here. Okay. From here, the ATP from this ATP, 1-phosphate was added here, and from this ATP, phosphate has been added here. This molecule is known as what? Fructose 1,6 bisphosphate. We call 1,6 biphosphate. Okay. Now there is another phosphate here. I have just shown with the P. Okay. This is that one. Okay. Now, in this, from these reactions 1, reaction 2, and reaction 3, we are investing the energy molecules. So this is known as what? Preparatory phase. What? Preparatory phase. We have invested. If you have to get a bulk of product, for example, you sow cotton or you sow wheat, first you have to plow, you use the energy, you invest, you use the fertilizer, you invest. That is the preparatory phase. And you sow the seeds of wheat or cotton, whatever you want to sow, and then it grows. You utilize water, you utilize again the fertilizer, you are investing. That is what the preparatory phase. And then payoff phase comes when the when it becomes ready for what getting the product. Okay, now this fructose 1,6 bus phosphate is now ready to pay back. Okay, now let us see how it does pay back. I would like to go further. Okay, if you have understood up to here, I would like to go further. This was all the preparatory phase. Okay, now we are going to start with what the payoff phase. Okay, the next reaction is carried out by an enzyme, and that enzyme is what aldolase. It cleaves this molecule into 1 2 3 1 2 3, and what happens if I show to this side? Aldolase has acted upon aldolase. Okay, and two molecules are generated. One is what? Glyceraldehyde 3-phosphate, and the other is what? Other is dihydroxy acetone phosphate. This is dihydroxy acetone phosphate. Okay, here is glyceraldehyde 3-phosphate. That molecule, this 6-carbon compound has been cleaved, has been broken down into these 2 3-carbon compounds. Okay, these are what? 1 2 3 1 2 3. So we call it what? Glyceraldehyde 3-phosphate. Okay, well, I may tell you people that these two molecules are interconvertible according to the need of the cell. Okay, here in the further reaction, what happens? Glyceraldehyde 3-phosphate undergoes the further reaction and forms what? 1,3 biphospho-glyceric acid. What? 1,3 biphospho-glycerate. Here is what? 1,3 biphospho 1,3 biphospho-glycerate. Okay, where this phosphate has come from, we have to answer to this question. It comes from an inorganic phosphate. Okay, and here what happens? NAD is reduced to NADH, and one hydrogen ion is also liberated along with that. In some other lecture, I told you people how NAD with plus sign means oxidized form that is reduced. Okay, you can search that lecture on YouTube, and you will find what's the mechanism of formation of NADH. Okay, here this 1,3-biphosphogylicerate has been formed. From 1,3-biphosphogylicerate, what is formed? 2-phosphoglycerate is formed. What? 2-phosphoglycerate. 1,3-biphosphoglycerate undergoes the biochemical reaction and forms 3-phosphoglycerate. And where is that phosphate? Here, ADP is converted into ATP. So here is what you got, the first ATP molecule, okay, in the payoff phase. In this payoff phase, you are getting now the energy molecules. This is again the energy packet. Okay. Here, the enzyme used is what? 1,3-biphosphoglycerate kinase. Okay. Now, from 3-phosphoglycerate, this is what? 3-phosphoglycerate. Phosphoglycerate. What is formed? From 3-phosphoglycerate, 2-phosphoglycerate is formed. I would like to remove this because you have already started this thing. From this, what happens? The mutation has occurred in the molecule. Phosphate has mutated from position 3 to position 2. So the enzyme used here is what? Mutase. Enzyme is what? Mutase. Okay. The actual name is what? Phosphoglycerate mutase. Okay. In the next reaction, what happens? In the next reaction, what is formed? See, phosphoenol pyruvate is formed. What? Phosphoenol pyruvate. Where have gone these two things? You understand? What? Water is released. Okay. And what next? In this reaction, again, the final ATP molecule is formed, and ADP is converted into what? ATP. Okay, and at the end you see pyruvate. What? Keto pyruvate is formed. This is 3-carbon compound. This is ATP. Okay, this is ATP. This is energy. This is 3-carbon compound. This is 3-carbon compound. 3 carbon, 3, 3. All these carbon compounds, these have been formed from glyceraldehyde 3-phosphate. What? This is what? Glyceraldehyde 3-phosphate. Okay. As I told you people, this glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, these are interconvertible. Okay. Dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, these are interconvertible. So this molecule will give rise to these energy packets, and at the end from this molecule pyruvate would be formed. So one pyruvate has been formed. What will happen next? This would be converted into glyceraldehyde 3-phosphate, and all these series of reactions would occur. Okay, and in this way, one ATP that would be doubled, 2, 2, and 2. Okay, so you get how many? 1, 2, 3, 4. 4 ATP molecules and 2 NAD. Okay, but I may remind you people that you have used in the cell 2 ATP molecules during the preparatory phase. So net ATP molecules are 2, okay, and 2 NAD molecules in the reduced form, which are the energy packets. These are also received at the end of what? Glycolysis. So this is how the glycolysis does occur in our cells. You have got the answer where the glycolysis occurs and how the reactions of glycolysis to occur. I have tried to tell you people where each hydrogen is used and how all the reactions are completed. Thank you very much. In the next lecture, I'll tell you people where this pyruvate molecule is used. Thank you very much.